Good afternoon, everybody. Today we are going to discuss the important topic of chemistry, that is atomic absorption spectroscopy. Generally, these absorption spectroscopy depend upon the absorption of photolight. Whenever there is a radiation takes place which is impart on the atom or molecule, then it goes in a higher excited state, and during that excitation it will emit some radiation and again come to the ground state. During these there is a absorption of energy takes place, and this amount of absorption has been calculated. These amount of absorption has been utilized for getting the signals from the which are detected by the detector and which is read out by the readout system, and thus these atomic absorption spectroscopy play a very vital role for the determination of different elements in the from the any mixture or generally from the ores. Therefore, this principle has been we are going to learn what is meant by atomic absorption spectroscopy. The basic principle of atomic absorption spectroscopy is that it determines or it measures the concentrations of elements, that it makes the use of absorption of light by these elements in order to measure their concentration. That means, we can able to calculate the concentration of unknown with the help of known also. Then the concentration measurements are usually determined from the working curve after the calibrating the instruments and with the standard of known concentrations. Then this is the periodic S and P block element as well as D block element of the periodic table. Among these elements, these whatever the red color I have shown here, pink color I have shown here, these elements are generally can able to detect by using a atomic absorption spectroscopy. Then the atomic absorption spectrophotometer has been contained the four important principles. First is the light source. These generally we are using the light source halo cathode lamp as a light source for these atomic absorption spectrophotometer. The second one is the atom cell, which is also called as the atomizer. The third one is the monochromator, and fourth one is the detector and the readout system. These four compartments are wholly considered as a atomic absorption spectrophotometer. Okay. Then this is a schematic diagram of AS. Generally, the initially the light source, which is halo cathode lamp. Then second one is the atomizer. Then third one is the monochromator, and fourth one is the detector and readout system. In this sequence, generally the arrangement of atomic absorption spectrophotometer has been carried out. Then this is the actual photograph of AS, which is available in the laboratory. Then first component of AS is the light source. Generally, the light source is usually halo cathode lamp of the element that being to be measured. That means it contains a tungsten anode and hollow cylindrical cathode made up of the elements to be determined. These are sealed in a glass filled inert gas. Generally, the inert gas are either neon or argon. Each element has its own unique lamp which must be used for the analysis. Then this is a halo cathode lamp. This is a picture of a halo cathode lamp. Generally, it contains the quad windows, then Pyrex body, then the anode. This is the anode, and this is a cathode. And in this way, this is the vertical photograph of our halo cathode lamp. Then working of AS, the applying the potential between the anode and the cathode, because halo cathode lamp, I already stated you that halo cathode lamp will consist of the anode as well as the cathode, and there is the potential difference between the anode and cathode that leads to the ionization of some gas atoms, and these gaseous atoms are bombard the cathode and eject the metal atoms from the cathode. In the process is called as sputtering. Some sputtered atoms are excited state emits the radiation characteristic of metal as they fall back ground to the fall back again on the ground state. Then the scheme of halo cathode lamp. Generally, the halo cathode lamp, there is anode is present, a cathode is present, as before in the previous picture I have already stated you the arrangement of a halo cathode lamp. Then atomizer, second component is the atomizer. Generally elements are analyzed needs to be in a atomic state. Atomization is a process in which the particles are break up into its constituent atoms. First of all, they are converted into a mist, then mist are converted into a anode and cathode, and then they are converted into its constituent atoms. There are two types of atomization are there, flame atomization and second one is a graphite atomization. Then this is the flame. This is the actual working of a flame for our atomic absorption spectroscopy. For example, from this arrow, the sample will be inserted or sample will become it is going to the for the burning flame. Then this is there is a light source is there, that light source, and there is a detector. So in this way sample is charged to the flame continuously, and the sample will convert into atomic form, and during that the excitation of atom has been takes place by the light source, and these excitation are read out by the detector, and in this way the working of flame has been executed. Then this is the flame atomizer. Actual work of that all the work has been carried out inside the flame, and so that is why the flame atomization technique is very much important in AS. These are the atoms in a flame. Then this is light source which is inserted during the through the flame. This is light source second, and through that there is atom are absorbed signal. That means atomic absorption signals are detected going to the monochromator, and through that monochromator its intensity will be increased, and after the readable intensity, then these are detected by the detector and go to the readout system. In this way the work of flame atomizer has been takes place. Then what are the different types of oxidant which are used in a flame? These are generally gas air. It has a lowest temperature range of 1700 to 1900. Then the maximum is the N2O and ethylene, and it has a range of 2600 to 2800 degree Celsius. Then this is the process of takes place in the flame, or how the process has been carried out there, in which first step, as I pictorially shown you in a previous slide, that solution of the analyte is charged to the flame, then in the form of a spray, then they are in the whenever the sprays go inside the flame, then there is the formation of aerosol particles, and these aerosol particles are converted into a gaseous molecule followed by the it constituent atoms, and these atoms are go in excited state by ionization and by volatilization. Then these excited atoms are converted into a atomic form, and these atoms, intensity of atoms, are useful for our determination. Then the effect of temperature on a excited state of population. Using this equation, it is a Boltzmann constant, and by using this equation, Boltzmann equation, we can able to find out the excited state of population. This is another form of these, and this is the sample introduction system through the atomic absorption spectrophotometer. Then this is the monochromator. Monochromator important part of atomic absorption spectrophotometer. It is separate out the thousands of lines. Without the good monochromator, detection limits has been severely compromised. That means monochromator has played a very vital role for the detection limits of the AS. Then this is detector and readout system. Generally the role of detector is to detect the signals which are given from the atomic form of the sample molecule. Then these readout by using the processing of electrical signal is fulfilled by a signal amplifier, and the signal could be displayed for the readout system. And then calibration, by using the calibration curve we can able to determine the concentration of unknown. This is the calibration curve, and by the using the slope of these, concentration has been calculated. then interferences, different problems, how the interferences can be resolved, and this is all about the atomic absorption spectrophotometer. I think that you maybe like these video for the your sake of knowledge. Thank you to all of.